Assalamu alaikum everyone. Our topic for today is the control of gene expression. So let's get into it. In order to understand the gene expression or the control of it, first we need to know what is a gene. Then we need to know what is gene expression. And then finally we can come to our topic, that is control of gene expression. So what is a gene? In order to understand what is a gene, you need to have the concept of what is a chromosome. And in order to understand both of these two terms, you need to know the basic cell structure. So a basic cell structure consists of, you have a cell, as I've shown right here, and that cell has a structure that is called a nucleus, as you can see over here. That nucleus contains chromosomes, and this we know from our prior knowledge that we have 46 chromosomes. Right? Now these chromosomes are actually structures that contain genetic information, and this genetic information is carried in the form of genes. In the form of genes. Right? Now what is a gene? A gene is a sequence of nucleotides, right? And DNA is what is made when these nucleotides align. So if we read the bookish definition, a gene is the basic unit of heredity, we know that already, and a sequence of nucleotides in DNA that encodes the synthesis of a gene product. Now that gene product could either be an RNA or a protein. So we have a cell, that cell has a nucleus, and nucleus contains chromosomes. These chromosomes are actually the structures that contains genetic information in the form of genes. Moving forward, we've talked about gene. Now how can a gene be expressed so that it can show its function in the body, or so that its phenotype can be displayed? I hope that you all know from your prior knowledge that you have a genotype. Why isn't it working? Yeah. You have a genotype and you have a phenotype. So a genotype is what your genes tell you. And what those genes can tell, that is actually the sequence of the nucleotides. It could be A, T, G, U, C. All of these are the nucleotides. And what is the phenotype? The phenotype is actually what is expressed, what we can see visibly. For example, if a person has, just as an example, if a person has this DNA encoding, then that person would have blue eyes. So we can know this, that a blue-eyed person has this genotype within the person. Now talking about gene expression, what is gene expression? It is basically how the gene can get turned on, or how the gene can actually start its process for what it's meant to do eventually. So gene expression is actually how the gene is going to affect the phenotype, how it is going to be seen visibly by us. And what is the process of it? From the chromosomes or from the gene, it is encoded into mRNA, and this process is called the process of transcription. And then from the mRNA, it is converted into the tRNA, or the process of formation of proteins. And this, when the tRNA goes and forms the proteins, because the tRNA is the molecule that contains the amino acids, this process is basically called the process of translation, because the gene is actually being translated into the required protein that it was meant to form within the human body. Right? So you can understand it from this paragraph. During gene expression, the DNA is first copied into RNA. The DNA is copied into RNA, the mRNA. The RNA can be directly functional, the mRNA can directly produce its effects, or it can or be the intermediate template for a protein that performs the function. Then the mRNA would form the tRNA, and the tRNA is going to form the protein. So this, in the case of the protein formation, would just act as a template so that the protein can be formed in the right sequence, the right amino acids, with the right amino acids. Then you need to know what is the difference between what are the different types of genes that we have in a body. So we have the structural genes and we have the regulatory genes. Talking about the structural genes, these are those genes that code for a protein, for a specific protein, and that specific protein has a function. And what is a regulatory gene? The name defines both of these genes themselves. The regulatory gene is a gene that codes for a protein that helps control the expression of the other genes. Now, the regulatory gene is basically controlling the expression of those genes that are going to produce a protein that has a function inside the body. So the regulatory genes are the genes that are regulating or controlling which genes? The structural genes. And what are the structural genes? These are those genes that produce the protein that has an actual function within the body. Then another very important term is the operon. So what is the operon? It is a unit that is made up of linked genes which is thought to regulate other genes. Now, I'll be giving the example ahead in using a diagram, but just in order to understand the word itself, we are going to go through these bookish definitions. So, operon basically consists of a number of genes that include the regulatory genes as well as the structural genes. For instance, if I give you an example, we have gene 1, 2, 3 and 5, let's say. So, I say that gene 1, 2 and 3 are regulatory genes, and gene 5 is a structural gene that is going to form a protein named X. Right? Now, we have given the name to all of these 4 genes. This complete structure is basically called an operon. Right? So, I have gene 1, I have gene 2, I have gene 3, and then with it I have a gene 5, right? And then apart from that I have the gene, some other genes, like another set of genes, right? So the structure, so this specific structure within the chromosome, is called the operon, that consists of the regulatory genes that are 1, 3, 5 and the structural gene, right? So you can have a look over here. Using this definition, structural and regulatory genes that work together are generally found in a group, and this cluster of genes is called an operon. I hope that you understand it. Another important thing that you need to know is this. The functional unit of transcription is called an operon, and the cluster of genes that are controlled by a same promoter. Now, what is a promoter? Promoter is basically the area behind the operon that actually allows the regulatory genes and the structural genes to be activated. If the promoter has a repressor on it, then these genes are not going to get activated, they're not going to produce the proteins. The regulatory genes are not going to regulate, the structural gene is not going to produce the required protein. So the promoter not only controls the regulatory gene, it also controls the structural gene, it also controls the regulatory gene. The promoter controls the operon. I hope that you understand this. Let me simplify it just for the sake, in case you did not understand. Just a second. Right. So quickly, I'll just recap this because it is very important. What is an operon? Operon is the cluster of genes that consist of both the structural genes and the regulatory genes. So this is a chromosome, for example. We have gene 1, 2, 3 and 5. Then we have another set of genes called 7, 3, 10, 15. Right? Now, 1, 3 and 5, these are the regulatory genes. 1, 2 and 3 are the regulatory genes and 5 is the structural gene. The whole unit that consists of the regulatory and the structural genes is called the operon. And behind these genes, the regulatory and the structural genes, we have a unit that is called the promoter unit. And this promoter is going to either promote the transcription and translation of genes 1, 2, 3 and 5, or it is going to inhibit the transcription and translation of genes 1, 2, 3 and 5. It is either going to allow the protein to be made, the promoter is either going to allow the protein to be made, or it is going to prevent the protein to be formed, from being formed. So this is the basic thing